Respected Dr. Ishwar V. Basavaraddi sir, Director Murarji Desai Institute of Yoga, to say a few words about today's topic on role of traditional medicine in prevention of dengue infection. Sir.
Shanti, Shanti, Shanti. Today's esteemed speaker, Dr. K.P. Mishra ji, a very prominent scientist from DRDO and DIPAS, and my colleagues in the institute, Dr. Tayabalam ji, Dr. Vandana, faculty members, student friends, and yoga enthusiasts joining these webinars from various social media platforms of Morarji Desai National Institute of Yoga. Today, I am very happy to once again welcome Dr. K.P. Mishra ji, who is working as a scientist of a very senior scientist of DRDO, Defense Institute of Physiology and Allied Sciences, which is popularly known as DIPAS. So you all know that both DIPAS and MDNIY has a long association during the last more than 3 decades or 4 decades, 1970s. So continuing that very profound association, today we have with us Professor Mishra, and he is going to enlighten us on the role of traditional medicine in prevention of dengue infection. Whenever the drugless systems like yoga comes, we talk about only lifestyle and stress management, non-communicable disorders, etc. But very few people integrate plant-based medicine, the modern medicine, and also combined with yoga and other drugless systems to combat the infections due to some insects or like this in coronavirus. We are not able to do directly, but that will definitely help to the integration of yoga with plant-based medicine and also other medicines. The yoga system will definitely help. As you are aware, India, including other South Asian countries, are very much affected by dengue fever. Every year, these dengue fever are causing several thousands of deaths or mortality across the globe in different parts of the world. In this lecture, Dr. Mishra will enlighten us about the vectors of dengue virus, how the spread of mosquito can be prevented, and how the virus infection leads to immunopathogenesis. That is very important, and he is going to emphasize on these 2 important aspects. Dr. Mishra will also enlighten us on the importance of traditional medicine plants, such as rhodiola, which is in native of Ladakh, and papaya leaf extracts, on which Professor Mishra has done some research. And these 2 medicinal plants, that is rhodiola, I pronounce it correctly, I think so, otherwise Professor Mishra will correct this, and papaya leaf extracts of 2 medicinal plants, has proven their role in the combating dengue infection which will boost our immunity and suppressing the virus replications in our body. So it's also important to know that some food intakes in terms of medicinal foods will also proven to be very important, and pranayama, particularly Shitali pranayama, Brahmari pranayama, and research show that they will be adjunctive in combating the very severe infection when the dengue fever is in the air side. I do hope so. It's very interesting for me also to listen to Professor Mishra. With these few introductory words, I would like to invite Dr. Mishra to address us on the topic role of traditional medicine in the prevention of dengue infection. Dr. Mishra.
थैंक यू वेरी मच सर। आप सभी को मेरा सादर नमस्कार। रेस्पेक्टेड डायरेक्टर मोरारजी देसाई नेशनल इंस्टीट्यूट ऑफ योगा, फैकल्टी मेंबर्स, स्टूडेंट्स, ladies and gentlemen, thank you for inviting me to this weekly webinar and for giving me the opportunity to talk about role of traditional medicine in prevention of dengue infection. So as you know, dengue virus is basically a virus belonging to family Flaviviridae, which has 4 serotypes, that is dengue virus serotype 1, 2, 3 and 4. So nowadays in this ongoing pandemic, you might have heard the name variant, different variants of coronavirus. So there is difference between the variant and the serotypes. So serotypes is generally they differently interact with the antibody present in our blood. So dengue virus has 4 serotypes, and these viruses serotypes are actually causing the antibody dependent enhancement phenomenon, which I will discuss in later slides. If I tell you about the genome of this virus, it is a positive polarity single strand RNA virus, which has 3 structural and 7 non-structural proteins, which primarily infects in our blood, which are the monocytes, macrophages, and dendritic cells. These are the immune cells, which are responsible for the guarding our body from the enemies like viruses, bacteria, and other opportunistic infections. This virus found throughout the tropics. If you see the world map, you will find that in the tropical climate, this virus infection occurs, and the infectious virus live in the stomach of the Aedes mosquito. So therefore, the virus transmission occurs by the bite of infected Aedes mosquito, and there are 2 types of Aedes mosquito. One is the Aedes aegypti, and second one is the Aedes albopictus. But primarily, it is the Aedes aegypti which majorly causing the infection. If you see the epidemiology of dengue illnesses, around 50 to 100 million illnesses occurring annually, out of which 20 to 25 lakh cases of severe form of dengue fever. So if I summarize, it can say that around 80% dengue fever is febrile in nature, but sometimes 20% cases become severe, which is known as the dengue hemorrhagic fever and dengue shock syndrome, and that may lead to the mortality as well. So here I have shown the immature non-infectious virus and the mature virus which is causing the infections, and its genome. So this is the world map where you can see the tropical countries or tropical climatic zone is actually affected with the dengue virus infection, and more than 100 countries of the world is affected with the dengue, and many more are uprising because of the air travel and because of the different types of movement where one can go, and therefore the uprising the dengue virus infections in those areas where the dengue was not reported earlier. So if I tell you the history of dengue virus, when the first time virus was isolated, It was the year 1943 when two Japanese scientists, Ren Kimura and Susumu Hata, who first isolated the dengue virus serotype 1. And 1 year later, Professor Savin isolated the same dengue virus, and later on, the dengue virus serotype 2, serotype 3, and serotype 4 were discovered isolated. This is the national scenario of dengue virus infection. Here you can see mostly the 16 states of India is endemic for the dengue virus infection. And these are the other: Andhra Pradesh, Goa, Gujarat, Haryana, Karnataka, Kerala, Madhya Pradesh, Maharashtra, Punjab, Rajasthan, Tamil Nadu, Uttar Pradesh, West Bengal, and some Indian territories, also Chandigarh, Delhi, and Puducherry. They are also affected with the endemic of the dengue virus. The first time in India, the dengue hemorrhagic fever epidemic was reported from Calcutta during July 1963 to April 1964. And if you see our own city, New Delhi, the outbreak of dengue fever reported in year 1967, then 70, then 1982, then 1996, then 2006, and after that 2013. So these, if you can see, the virus outbreak occurs nearly after 6 to 7 years gap. Now, as I said, this virus harbors in the stomach of the mosquito Aedes, and here I have tested these two, the mosquito Aedes aegypti and Aedes albopictus. And you can differentiate between these two mosquitoes by looking at the thorax region of this mosquito. The Aedes aegypti is having the sputum, which is layer-shaped structure. The layer looks like this bell-shaped structure over here, and this is a white marking. It is observed in the thorax region of this Aedes mosquito. While in case of the Aedes albopictus, there is a long, medium longitudinal white stripe extending from the anterior region to the base of the wings. And there are white patches on the legs, as you can see over here. So this is clear-cut indication, demarcation of the Aedes aegypti and Aedes albopictus. Now, a virus, mosquito, when bite, it pours the virus inside the dermis of the human being. So mosquito in 10 days become an adult mosquito. It lays the eggs, millions of eggs, stagnant fresh water, and within 2 to 3 days, it become larvae, and then it become pupae, which require 4 to 5 days. And from pupae to adult mosquito, it takes only 2 days. So if you see total time period of a mosquito, you will find that only 10 days a mosquito can be able to produce millions of the new mosquito ready for the virus infection. So it is very important to prevent the mosquito breeding in our vicinity and in our societies where we are living, because it always lay the eggs in the stagnant water. Now, coming to the immunopathogenesis of dengue virus infection, this is a mosquito which is biting skin, human beings' skin, and you can see the skin is divided into 2 layers, epidermis and dermis. It pours the virus particle in the dermis, where our own innate immune guards are present, and these guards try to neutralize this virus as soon as they sense that some kind of virus or foreign particles came in our body. So these cells which are the sentinels of our immune systems, are the macrophages and dendritic cells, and also the natural killer cells. But sometimes dengue virus escapes from these guards, and it goes into the blood vessel, where it causes it reproduces and makes its population very high, and then a severe form, the viremia phase occurs, where the fever response comes. So after biting of a mosquito, 4 days later, you will feel fever. So during the onset of fever, one can detect whether he is infected or not infected with the dengue by 2 techniques. One is the ELISA technique for NS1 antigen, and other one is the real PCR for RNA analysis, the RNA presence of these viruses. So viremia phase occurs for the 5 to 6 days, and then after viremia, there is a severe form, or you can say the critical phase, that is characterized as the dengue shock syndrome or dengue hemorrhagic fever. Here the bleeding occurs from nose, from eyes, even from in the gut also the bleeding occurs. And the most severe form is the dengue shock syndrome, where the hypovolemic shock occurs, the blood pressure get down, and then it become very critical phase. Here, these 2-3 days are very important to manage the dengue fever. Now, coming to the life cycle of dengue virus, when the virus bind to the receptor on the surface, and then receptor-mediated endocytosis occurs, and by low pH fusion, the virus take out its RNA in the cytoplasm, and using the host cell machinery, the viral RNA gets translated into different types of structural and non-structural proteins. As I said, there are only 10 proteins present in the virus. So after translation, the synthesis of viral RNA is known as the translation. So translated proteins then goes to the Golgi vesicle, and from Golgi vesicle, it goes to the membrane, where it fuses with the host cell membrane, and egresis occurs, means it bursts the virus, the cell membrane, and again ready to infect the another healthy cell in the vicinity. Now, unlike the coronavirus, you all know that coronavirus bind to the surface receptor known as the human ACE2. The ACE2 is the receptor which bind to the spike proteins of the coronavirus. But in case of dengue, there are many receptors, and therefore it is very difficult to target the one receptor. In case of dengue, you can see there are many receptors like beta, the heparan sulfate, manoj receptor, C-type lectin known as the DC sign, HSP70-90 receptor complex, TIM-TAM protein receptor complex, and one important receptor which is present on our cells is the Fc gamma receptor. It is very important to understand the antibody-dependent enhancement phenomenon. Here I would like to tell you that dengue virus has 4 serotypes, dengue 1, 2, 3, and 4. If suppose I have been infected with the dengue virus 1, I will produce the antibody against the dengue virus serotype 1. But suppose second time I am infected by dengue virus serotype 2, so my antibody for dengue virus 1 will facilitate the dengue virus serotype 2 into the cell. That means it makes more opportunity to get infected, the other cells to get infected, so causing the antibody dependent enhancement phenomenon. And this phenomenon only causes the dengue vaccine is not so far fully developed. We have also added some important knowledge in the understanding of dengue immunopathogenesis, that HSP70 and HSP60, these are the 2 host cell proteins which facilitate a virus to multiply inside the cell, which has been published in 2 prestigious journals like and Viral Immunology. Furthermore, we have also identified a protein known as the protein disulfide isomerase, which exists in the lipid wrap domain of the cell membrane of the host cell, which also facilitates the dengue virus entry inside the cell. I am going to tell you about this, how we have identified this protein specifically present in the host cell membrane, and how we can treat the virus infection or suppress the viral replication. So for this, to fulfillment of this objective, we have established an infection model using the human monocytic cell line, which is known as the THP1. This human monocytic cell line, we have standardized the dose of virus, which is of the multiplicity of infection of 2 for 72 hours of infection. And then we tested whether cells are infected or not infected using different modern approaches like the viral plaque assay, Western blotting, and RT-PCR. These are the molecular techniques and the available methods by which you can detect whether virus is present in the infected cell or not. Then we did 2-dimensional gel electrophoresis in the infected and compared it with the uninfected cells. What we found, there are 2 proteins which get upregulated. These are the heterogeneous repair ribonucleoprotein H and protein disulfide isomerase. And out of these 2, the protein disulfide isomerase were showing higher probability-based mouse score, which is 106. So we were interested why this protein disulfide isomerase get upregulated by virus infection. Does it have any role in the virus multiplication? Does it facilitate virus entry inside the cell? So what we used the RNA interference technology. This is a technology by which you can suppress any kind of gene. And for this technology, in the year 2006, Nobel Prize was awarded. So siRNA technology discoverer has given us a tool to suppress any kind of gene. So we have used this siRNA technology, and using this technology, we have suppressed the protein disulfide isomerase. And what we found when we suppressed the protein disulfide isomerase, the viral replication was decreased. Now the question was, which viral protein is actually interacting with the host cell protein PDI? So we have screened all the host, the viral proteins with the protein disulfide isomerase and identified that viral NS1 protein is actually interacting with the protein disulfide isomerase. And when we tested the localization of the PDI and NS1, we found that yes, protein disulfide and NS1 are closely reside. So that means the PDI and NS1 interacting with each other. Now, to know the role of protein disulfide isomerase in viral binding, we inhibited this protein cholesterol-rich domain in our cell membrane.